Kia ora, kia tau, everybody. So my name's Tara Murray, and I'm with Department of Conservation down in Dunedin these days. And I've been very interested, probably in about the last 7 years, trying to get my head around a little bit the invertebrate biodiversity values of our braided rivers. Once we have a sense of what's actually out there, then when building and all the stuff with the likes of Holly, with looking at food web stuff and how all these ecosystem processes are all linked. But I'm going to talk today about some studies we've been doing on the Ashley and Cass in particular. And my title there says, "And impacts of weeds and flooding," and small disclaimer that we don't really know yet. But I'll tell you a few little things that we've seen so far. So this work all stemmed from some work that was developed by Project River Recovery in the Twizel Dock Office back in 2005. They did a really intensive study of invertebrates using lots of different trapping methods over the entire season. And from that big study, I went in and looked at all that data and tried to figure out all the kind of what, when, where, why, and what to kind of sample across these braided rivers to get a sense of what the biodiversity values are, so that we could then go to other braided rivers and find out what the biodiversity values were in those as well, without doing quite so much work. So we went through that and came up with a recommended protocol for sampling. And over the last few years, I've been applying that to a couple of other river systems, particularly the Cass, Ashley, and Aparima. I'm mostly going to talk about the Cass and Ashley today, which is a study that is jointly worked on by DOC and ECAN. And one of the first things we want to do is test out these methods, see how good they are, get a sense of the baseline biodiversity values for their own sake, what's actually out there. And build the sampling in a way that we could get some understanding of some of the weed impacts on the invertebrate biodiversity on the rivers, and the weed management impacts. And as you'll see as I go through, we've morphed that a little bit into also impacts of flooding, because rivers do what they do. So the Lower Cass has just been introduced to you by a number of different speakers, so I don't have to really do it again. This is kind of our more pristine control site, if you like. Fairly natural flowing upland river, relatively weed free. So we only have 3 sites established in this location. The second one is the Ashley. This has we have 6 sites here, and we had set them up in a way where we had 3 of our sites in a relatively unmanaged part of the river. For weeds, and then we had another 3 sites in an area that had undergone this raking that I think you will know about if you've been coming to these seminars for a while. And I think we've had a few talks on this before. You can see the image there of the one of the various contraptions that they've used for raking out the weeds. So this was around removing weeds, over consecutive years and trying to get that seed bank down. If you want to know more about that, talk to Nick, because he's the expert. So we've got 3 unmanaged and 3 raked sites on that one. And then down south in the Aparima, we again have 6 sites, and they ranged a lot more around their weediness, quite a different river. This is the worst kind of site, fairly unnatural looking, that one. But this river, we set up our sites in a way that we intended to go back in the future to 3 sites where we're expecting gravel extraction to occur. So we're kind of gathering baseline data, and then we want to look at the effects of gravel extraction later on. And then 3 other sites where that process wouldn't happen. So I'm not going to really talk much about this particular river, just putting it in there to let you know that there's some other stuff going on there. Methodology-wise, so when I say site, each of our sites consists of a malaise trap and 5 pitfall traps. And in each of those sites, we go back and we sample 5 days and 5 nights, once a month from November through to February. We have a DOC casual staff who always does the Cass site. It's Jolene in the front. I'll go back to you there. She did it last year, and this year we actually had the Ashley-Rakahuri group running the Ashley site, which was fantastic because, as I said, I'm now in Dunedin, so it's quite hard for me to run. And there we are, all just learning how to actually set a decent trap to catch our bugs. I really appreciated the work from the Ashley-Rakahuri group this year, and here's Nick, and I think Judith. I have helped him with that particular one, but I have to say that is the nicest set up Malays trap I've seen from any volunteer ever. So I've asked them if they'll do this again next year. And there you can see the pitfall trap design. So a pitfall trap is a hole in the ground that things fall into, but it does have to be set well to actually catch bugs well. And you can see we've got these little arms there, which is to increase the surface area over which the insects are funneled in towards that trap. So at each site we have a Malays trap at the front here, big tent basically, which intercepts insects that get caught when they go up into the top of the trap. And in the background, hopefully you can see the line there of the pitfall traps extending back from it. You catch a lot of bugs, bring them back into the laboratory, and we've basically been processing them through anything over about 2 millimeters size. We've been identifying to something called RTU, or Recognisable Taxonomic Unit. This just basically means we try and work out how many different species we have, but we don't always know what their names are. So if we're lucky, RTU 43, for example, we actually know it's a particular carabid beetle species. We've got its genus and species name. RTU 99, we know what genus it is, but that's all. RTU 837, we know it's a carabid beetle, and it's different from the other carabid beetles, but we don't know what its actual species name is. So in terms of results, I am fairly early on in analysis here, so I can't really get into nitty-gritty detail, but I'll just give you an overview, really. We have, over the last 3 seasons so far, we've sorted 60,000 specimens, and these are just the big ones. That number's going to go up because we haven't finished our Malays samples yet. That's fairly standard for invertebrate work. You get lots of bugs and traps. And you can see here, we've just got the number, this is abundance, number of specimens in the different rivers. And you can see we get a lot more in the Malays samples than in the pitfalls. Malays intercepts stuff moving through the air. It's not necessarily insects that have grown up there. They could have come from miles away if it's a windy day, or they might have emerged right next to it. But it tells you kind of what food's moving through the environment. The pitfall stuff, smaller abundance, but some of those invertebrates, those are the invertebrates that are like really tied to that particular area, and they'll be food sources for some particular birds like dotterels. They'll be feeding on that terrestrial stuff. Yeah. We got over 1,000 species RTUs so far that we have identified, and they're from a whole bunch of different invertebrate orders, as you would expect. Mostly half of them insects, other non-insect arthropods like millipedes and centipedes and things like that. And then only in the Aporema we've also got the odd slugs and snails and even the odd worm. I just want to point out here the Cass. You can see this is our upland river. Numbers are always a little bit lower in our Cass samples. That's partially reflecting the fact that we only have 3 sites there rather than 6, and I haven't quite got around to adjusting my my data to show you the kind of per trap results yet. So in terms of the biodiversity that we've picked up so far, I just thought I'd have a really quick look at what's making up the biodiversity in the different rivers mostly by insect order. And here we've got just the Cass and the Ashley. And you can see that remarkably similar in terms of what we're picking up. So a good almost a third of the things that we're detecting are flies, different types of fly species. Then we've got a whole lot of wasps and a few of our native bees. Some moths, and then you get down to these smaller components here. that's where a lot of your kind of pitfall diversity comes in. You got more of your spiders and beetles and little creepy crawly things that are actually running around on the ground rather than flying around in the air. I put EPT in there for the aquatic interested people. So that's the Ephemeroptera, Plecoptera, Trichoptera grouped in there. And I just, out of curiosity, I thought, well, I wonder how this compares to the big Tasman study that we did, years back, where we got this really big river, very much more pristine environment. And I was quite surprised that the proportion of the diversity is actually panning out very, very similar. So that's pretty cool. What that doesn't tell me is what the species composition in each of those groups is. So they might be a completely different community. I don't know yet. I'll have to delve into the data. If there's one thing I want you to take away from this, it's that bugs are cool and we should look at them more. So I thought it's very easy to overlook. I'm preaching to the converted, I know, but it's very easy to overlook the biodiversity out there that's really little. So I'll just give you a snapshot of some of the cool critters out there. And as I said, about 30% of all the insects that we're picking up in our, particularly our Malaise traps, are different species of flies, and of course lots of birds and things are going to be feeding on these. A lot of them will be aquatic in their juvenile stages, so they're they're interacting in different parts of the trophic communities as well. So these are all different flies. Don't ask me what they are. These are a whole bunch of the beautiful wasps. Most of them will be parasitic wasps. One little native bee in the middle there. On the Tasman, we actually picked up something like 14 species of native bees. I've only got a couple in the Cass National. Some of our beautiful spiders. These are going to be a really interesting group to look at more closely because, of course, they're a top predator in the invertebrate sense. And these are some of the harvestmen or apilioines. So they're also arachnids. But, they range from predators through to scavengers. Some of our coleoptera are beetles. Beetles are fairly underrepresented in this environment, which isn't really surprising because beetles range across all the different kind of feeding habits from detritivores through to predators. Because you haven't got a huge diversity of plant life in these areas that we're sampling, you don't have a huge diversity of the herbivorous beetles. So we're getting quite a lot of these will be predators, and then you'll get a few that are generalist herbivores and omnivores as well. And these are just some of the sapsuckers, mostly some of the sapsuckers, so true bugs. All these ones on this side here, different sap-sucking bugs. And then we've got, this is just a little lacewing, a Psychoptera, it's common name, book lice, and some cute little snails. But that doesn't really tell much of a story yet. So, I mean, we know there's lots of diversity out there. We're picking up lots of stuff, but what does it all mean? I guess what I'm going to be looking into over the next wee while, and I've got one more year of sampling to do yet, is looking at things like these trends in abundance. This is the number of specimens caught, number of species detected with pitfall trapping in these sites. I'll be looking at those trends over the different rivers and over the different years and the different sites within rivers to try and understand the variation that we're seeing in the captures and the different species that we're picking up. So, what species are actually causing, for example, this big jump in the last season. We got a whole heap more specimens for some reason, and what is that? And what are they responding to? Just as an example, I looked at this, well, this Cass bar seems to have gotten very high very quickly. What's that all about? A very quick look at the data showed me, well, actually half of those are ants. So I need to go and cull that out from the data set because we all know you see one ant, then you'll see 1,000 and you'll just, they're not going to really tell you much about what's happening in the environment. But the other thing that makes up a lot of that extra specimen count that year was a small caddisfly, particular species of small caddisfly. And we saw a very similar thing a couple of years ago in the Aparima River. So I'm quite curious to go back and actually learn a little bit about what particular caddisfly is this, and what kind of things does it respond to? And the Ashley, again, we also got a bit of a peak this year in catches. And again, I went back and looked, and about half of that extra abundance was due to little chironomids. And they're a bit like ants. You get one, you get hundreds. So that might have just been that we had a really good emergence that week. So that probably isn't signalling anything. But what we also got, the other half of that increase, was beetles, a particular beetle, this little rabid beetle. And when I look at just the number of beetles caught in each of the different years in the two rivers, you can see that spike was really quite prominent in the Ashley for this year. So I need to go and have a look at, what's that little beetle potentially responding to, because beetles take a lot longer to actually develop. It's just not something that's mass emerged overnight. These have been growing probably over months, and then emerging as adult beetles that we've actually picked up. So as I said, I next need to start going to what are the drivers behind any changes or any differences in community composition that I'm going to be seeing here. I want to go back and have a look at those different years and the different sites, like the raked sites and the weedy sites and the Ashley in particular, and see if we're actually going to pick up, or if we're picking up different community compositions and changes over time. This is just something I pulled out of the first 2 years of data for the Ashley River, and it was quite exciting initially, because we're like, these sites in the light green were the raked sites, and then the dark green is the weedy or unraked sites. And we're like, oh, we're actually picking up more diversity in the raked sites, in both our Malays traps and in our pitfall traps. And then the next year, that difference had declined. And I was thinking, oh, maybe the raked sites, the weeds coming back, maybe the communities are converging back to being the same. And I was really looking forward to the third year of data to see how that all panned out. But of course, between the at the end of that sampling season and then again during this most recent sampling season, we've had multiple floods in the Ashley that have kind of made our raked and weedy sites fairly null and void. They're all fairly similar again now. So understanding what is the impacts of floods, and are we seeing any responses of the invertebrates to those flooding events? Grant Davey was kind enough to provide me with these images across the one set of the sites in the Ashley River. We can't get them at the other site because it's too close to the airport, so we can't get a drone up there. But you can see this is just the change in the channel flows over that sampling season when they were out there. At the three raked sites, the little red circles are the start points of each of the sampling lines, and you can actually see the Malays traps and the little white dots there in that first image. So over the season, you can see, just after we put them out, next month, you can see the weeds are starting to come up, the lupins are starting to cover bits of land a bit more. And then we got a couple of decent floods through there, and didn't quite lose our sampling lines, but some of them did go underwater at times. So we were interested to see how that might change what we're actually catching. And this is one of those particular sites before the flooding. You can see the three Malays traps there, and this is, I think it's this trap here, should be about here, during that particular February flood. And we actually had to delay the February sample into early March to let the water recede enough for us to even get to where we wanted to put the traps back out. And I had a quick look at the data to see, well, gosh, did we even catch anything after that event? And here we've got, again, abundance and diversity over the different months and over the different years and the colours there. So there was a flood here just before the February sample, so 2-3 weeks before the February sample. And I was thinking maybe those numbers are going to be rock bottom. We're not going to be catching much, but as you can see, we actually got more specimens and more diversity captured 3 weeks after that flood than we had in the previous couple of years on the same month. So, you know, Nick was asking a question about how do these things respond, how quickly do things come back. And of course, these are the terrestrial life stages of these invertebrates, or fully terrestrial invertebrates in some cases. So some of these individuals, their habitat was completely underwater, so they've come back in from somewhere during just those 2-3 weeks after that flooding event. So next steps, I guess I want to point out that we now have, we've started to build a really rich, interesting data set that we can mine in different ways to look at these invertebrate communities, and hopefully in the next few years I can come back to you and actually tell you something about the community structure rather than just how many things we caught. This, I think, highlights this dynamic nature of braided rivers. We know they're dynamic. It's been really interesting to see that this study thing that we wanted to do on weeds has been hampered by this, you know, these flooding events coming through and actually resetting the weed loads in those particular sites. And there's nothing much we can do about it. We just need to work around it. Having sample over these multiple years, though, is really useful because it's going to help us understand a bit more about the fluctuation that we see over time and space in the invertebrate captures that we get, and help us learn to actually interpret that data. Because at this stage, it's really easy to catch bugs, but interpreting what they actually mean functionally in terms of, you know, their own importance, but also functionally in terms of, you know, the food value that they provide to other organisms in the environment. That changes all the time, and how does that respond to different events in the environment? So we'll be looking at community composition, functional variation, and hopefully teasing out any information I can around the weeds and the flooding events that have occurred. So that's it. I just want to thank again the Ashley-Rakahuri group for their work out there this year, Jolene, who did the sampling this year, Jennifer and Sam, who aren't here today, unfortunately, for doing the sampling the previous couple of years, and Jessica Chen, who's been doing all the bug picking multiple years now. Cheers.